In this session, we'll talk about nutritional requirements of the eggplant, particularly organic. We know that plant need different type of the nutrients. Out of that, organic substances are also one of the important component which is required by the plant under in vitro condition as well as in vivo condition. Already we have discussed about inorganic component. Now we are going to talk about the organic substances. So these organic substances can be divided into two parts. One is a carbon source, so which we are going to provide in the form of the sucrose, and another is vitamins and amino acids. So vitamins like thiamine, nicotinic acid, so these vitamins are required or we are going to add into the media, and amino acids like glycine we are going to add in our media. So these organic substances particularly, will talk first about the carbon source, that is the sucrose. So we plant cell, what we are going to inoculate, what we say the eggplant is a small part of the plant which we are going to inoculate on the media. So it is not able to do the photosynthesis, means it is lacking the capacity, or it is a lacking capacity of photosynthesis, means it is lacking the autotrophic ability, and because of that we need to externally provide sucrose as a carbon source. Because when we are going to autoclave our media, so this sucrose will be broken down into the glucose and fructose, and out of that glucose will be utilized by this plant. When the glucose will be finished, then later on plant will start to utilize what we say the fructose. So we are going to add into our media sucrose as a carbon source. So another organic substance which is added to our media is vitamins. So vitamins are added into the media because they are required for the various metabolic functions, or they are going to catalyze certain type of the reactions in the plant body, and because of that they need to be added. Another reason is that these vitamins are synthesized in the plant body endogenously, but they are synthesized in the suboptimal quantity, and because of that we need to add these vitamins to our media. So when we are going to prepare our media, we are going to add different vitamins like thiamine, that is a vitamin B1, nicotinic acid, that is vitamin B3, and pyridoxine HCl, what we say the vitamin B6. In addition to that, myo-inositol, which is also vitamin, is also added into our MS medium. So the next compound is amino acids. Particularly amino acids like glycine, glutamine, asparagine are added as a nitrogen and nitrogen base like adenine are used as a additives in tissue culture media. These are added in this very small quantity to the media, and that will help the plant for their growth and development. Particularly when we talk about the MS media, glycine is the most common amino acid which is used in different culture media. In MS media, this glycine is particularly used. It is not essential, but nitrogen containing amino acid enhances growth, and because of that during the plant generation amino acids are added. Particularly in our MS media we are going to use glycine is added to the media. So when we talk about the different type of the compounds, the next compound which is added to the media, which is again having a organic which is also organic compound, is a growth regulators. So there are different type of the growth regulators are there. So auxin, cytokinin, gibberellic acid, what we say the abscisic acid, and ethylene, these are the 5 basic phytohormones which are found in the nature. So in addition to that, there are certain compounds which are having this type of the activity that may be also added into the media as per our need. So when we are going to talk about these hormones, particularly we will talk about these plant growth regulators, auxin and cytokinin in detail, because they are basically required for the particularly this in vitro type of the growth and development. These phytohormones are going to play a very important role. So what kind of the roles they are playing, so we will discuss first, and then we will talk about auxin and cytokinin. A plant growth hormone can be defined as a small organic molecule that actually accelerates physiological responses even when they are present at very low concentration. So such a low concentration is going to accelerate physiological functions or responses of the plant, are known as what we say the plant hormones, and these plant hormones are synthesized by the plant. So they play a very key role in the what we say the growth and development of the plant. They acts as a messenger between the environmental conditions and what we say the genome. So if there will be change in the environmental condition, so they respond and they give signal, and accordingly they react. So the plant will give response to that condition. For example, I will tell you about one growth regulator, that is the abscisic acid. So see, when plant is going to transfer from what we say the towards the winter, when season is going to be changed, during that time plant is going to accumulate this abscisic acid in large amount, so that the growth will be arrested by this abscisic acid. So this is in response to the what we say the photo period, because photo period is going to decrease during that time. So likewise, so it is going to play. So other what we say the hormones are there, they are playing also very important role. But remember that particularly auxin and cytokinin are added into our media. So auxin, we will talk about this, because this auxin, as I have already told you in the previous lecture, if you remember, so auxin was discovered by Dutch scientist Wendt in 1926. So this hormone is very important, and it is responsible for the cell elongation particularly. So cell elongation is an important function of this auxin, as well as it will induce the unorganized mass, or it will induce the formation of the cells. Okay, so unorganized mass of the cells known as a callus. It will also help in the more quantity when auxin is added to our media. It will develop it will accelerate development of the roots as well as embryogenesis. What we say from the somatic cell embryogenesis development, so that will be induced by this auxin, and because of that natural forms of the auxin like IA or synthetic forms of the auxin like NA or 2,4-D are used in our media. So they play a very key role in the growth and development, and because of that in the various concentration we need to use them. The next hormone, you will be able to see the hormone cytokinin. So this is also one of the key hormone required in the growth and development of the plant. So auxin is going to accelerate the elongation of the cell. So cell will be elongated, and it is going to happen because cell wall plasticity will be induced by this auxin. And the next part, what we say the cell division, so that will be induced by the cytokinins, remember, and because of that in the cell cycle it is going to play very key role. So this cytokinin, there are many different forms are there, like natural and synthetic forms are there. So synthetic form is a 2IP, which is the most active cytokinin, and like that natural forms of the cytokinin are there, kinetin are there. So these hormones are going to play key role. So in the cell division, not only that, when we talk about the natural hormones like BAP and kinetin, they are going to, or all cytokinins are going to induce what we say the shoot formation. So this is the important function of the cytokinin. So auxins are assigned for the root induction, and cytokinins are assigned for the what we say the shoot induction. So this is going to happen under in vitro condition when we are going to use these hormones. Likewise, there are other hormones are there, gibberellins are there, or what we say the gibberellic acid. So this is the best example of that. It is rarely used in our plant tissue culture media, and because of that it is not used regularly in our media. So it is also going to do the elongation of the shoot. It also induce the what we say the flowering. There are so many other roles are there, but it is rarely used in our media, remember. Very few people have used this hormone in a media. So the next hormone is the abscisic acid. As I have already told you, it is also rarely used. But when you are going to induce the somatic embryogenesis, and if you want to arrest the growth of the somatic embryogenesis, so generally this abscisic acid is used, remember. So when the in case of the in vivo grown plants, you will find that what we say the maturing seeds are there, so they are not going to be germinate, though there is a lot of water is there. So immature seeds, there is an embryo, there is sufficient amount of the water, even though they are not going to germinate because of the presence of this abscisic acid. So to do similar role, we are going to add abscisic acid into our media, right, to arrest the growth of the somatic embryoids. So we'll talk about this in the another session or another lecture. So we will talk about the somatic embryogenesis. So the next, what we say, the required compound in the growth media is a gelling agent. So gelling agent like agar or agarose are used. So it depends upon the need of the user, right? Next, when we talk about these gelling agents, so these gelling agents are there for the gelling, or some people say solidification, but it is not, remember, solidification, it is a gelling process. So to give the support, what we say, our medium when we are going to prepare, so we are going to use the solutions of the different salt solutions like inorganic salts, like macro salts, like micro salts. Then we are going to dissolve the sucrose, we are going to add that, we are going to use the vitamin and amino acid solutions, we are going to use the growth hormones which are in the solution form. So we need to maintain our culture. So we need to sometimes, depending upon the need, so because if you are going to use that liquid media, so you will go for the suspension culture. So if you want to make it as a what we say, the medium, like gelling medium or solidify medium, so then you need to use compounds like agar, agarose or phytagel like type of the compounds. So different type of the gelling agents which are available in the market, so that can be used for this gelling process. Naturally occurring product agar-agar, which is derived from the red algae Giladium gracilaria, so that can be used as a gelling agent. Because of this addition of the this gelling agent agar-agar, so when you are going to add, generally in the plant tissue culture, it is used in between like 0.8% to 1% agar-agar is added into the media. Particularly 0.8% is the very standard quantity. So when you are going to add per liter, remember, so when it is added and when the solution is heated to 100 degrees Celsius, when it will get dissolved, and when the temperature will decrease to 45 degrees Celsius, it will get solidified or the gel will be formed. So it will give support to our plant material. So in case of the liquid solution, you need what we say, the shakers, so that thing would be not necessary here in this type of the culture. So solid media, if you want to prepare, you need to use this agar-agar. Agarose is another form of the, what we say, or the very purified form of the agar. So that can also be used as a gelling agent. So which melts above the like 30 degrees Celsius, and below that it is going to be solidified. The next one, gelling agent nowadays is available in the market, is a gelrite or phytagel. Gelrite is a naturally derived polymer and produced by the microorganisms, particularly bacteria, Pseudomonas, Elodia. So this bacteria is going to produce this polymer, and that is used as a gelling agent. It is a low cost, and 0.1 to 0.2% concentration per liter is required. So because of that, it is one of the best gelling agent nowadays people are using. So one of the important thing, so when we are talking about the media, so the media, pH of the media will be very important. And remember that for the better growth and development of our plant or of our X plant, we need to maintain certain pH, and that pH must be adjusted between 5.6 to 5.8. And this should be done before gelling and sterilization, remember. So before addition of the agar-agar, you need to add the adjust the pH with the help of sodium hydroxide, that is NaOH, or potassium hydroxide, that is KOH, using this solution. So if you want to increase pH, you can increase the pH by using these solutions. Remember that dilute solution of this, or if you want to decrease the pH, you can use what we say, the diluted hydrochloric acid, and you can adjust the pH. If pH goes below 5, means it will be more acidic. So in that case, gelling is not going to occur properly, remember. Or if the pH is going to be above pH 6, so if it will be in the like basic range, so the medium will be more hard. So you have to remember that. So anytime if you are going to prepare media, and if you are not going to you see that gelling, so that may be because of the low pH, and if the hard media is formed, it means that it is because of the what we say, the high pH. So remember that this is a very important thing regarding the media. So that is otherwise your plant will be not able to grow in that low pH or very high pH. Remember that.